Today we're going to continue talking about protein synthesis, and this is actually part 2. In this video, I want to explain how mRNA can be translated into a protein. Okay, so just a quick recap from part 1. Protein synthesis has 2 steps. Step 1 is called transcription. That's where you go from DNA to RNA. And step 2 is called translation. That's where you go from RNA to a protein. So in the last video, we discussed step 1. But in this video, in the remainder of this video, I want to discuss step 2. Okay. At the end of transcription, let's pick up where we left off. At the end of transcription, we had a piece of mRNA, which stands for messenger RNA, and it looks like this. That mRNA leaves the nucleus and it carries the message. That's why we call it messenger RNA. It carries the message that was stored in the DNA. That message was the instructions on how to build a protein. It carries that message to the ribosome, where translation will take place. So what is the ribosome? The ribosome is an organelle in the cell where proteins are made. And the ribosome is made out of rRNA. That rRNA stands for ribosomal RNA. And it's also made of protein. So it's made out of a combination of a type of RNA and protein. The ribosome has 2 parts. It's got a large subunit and then a small subunit. So the large one's on top and the small one's on bottom here. Okay, so the ribosome is where translation will take place, but what exactly is translation? Well, when we ordinarily speak of translation, we mean something like this. Translation happens when you convert a message in one language to another language. Now, during translation, the message in the mRNA is going to get converted from the language of nucleotides into the language of amino acids. Nucleotides are the monomer of nucleic acids. Remember, nucleic acids is what the Na stands for. But amino acids are the monomer of proteins. So we've got 2 very different things here. We've got nucleic acids on the one hand, and we've got proteins on the other. And converting the message from one into the other is kind of like going in from one language to another. That's why we use this term translation. Okay, so big idea. If you didn't hear anything else, hear me say this. Translation is where we go from RNA to a protein. Okay, now there's going to be some specific steps, and we're about to get into that. But first, you need to know about something called tRNA. So in order to do translation, the cell uses tRNA, and that T stands for transfer RNA. The tRNA has an amino acid at the top, and it has 3 RNA bases at the bottom. And those 3 bases at the bottom, those are called the anticodon. And you'll see why it's named that in a moment. Now, why is this called tRNA or transfer RNA? It's called transfer RNA because it transfers or transports amino acids to the ribosome. The tRNA is kind of like a little delivery truck in the cell, and it's going to pick up amino acids and bring them to the ribosome, just like a delivery truck would pick up some things and deliver them. Okay, so that's tRNA, and the cell has to have tRNA in order to do translation. Okay, now we want to get into the specific steps of translation. So there's going to be 4 basic steps of translation. First, the ribosome attaches to the mRNA. So our mRNA got made during transcription. Remember, that was step 1 of protein synthesis. Now we're on to step 2 of protein synthesis, which is translation. And the very first thing you're going to do in translation is have the ribosome attached to the mRNA. Now, the second step of translation, a tRNA that's carrying an amino acid binds its anticodon. Remember those 3 letters at the bottom of it. It's going to bind its anticodon to the codon on the mRNA. So the mRNA, every 3 letters on the mRNA is considered a codon. So AUG represents the first codon on this mRNA. The second codon would be GGC. The third codon would be AGU. So you can see every 3 letters on the mRNA represents a codon. The tRNA that has an anticodon that complements the codon binds. That's what happens in this step of translation, this second step of translation. Now, the rule, the base pairing rule between the codon and anticodon is like this. An A will pair up with a U. So you can see the A on my mRNA binds with a U on the tRNA. A pairs with U, and G pairs with C. So I have a C and a G together here. The G on the codon binds with a C on the anticodon. Okay, so that's the second step of translation. Now, the third step, a bond is going to form between the amino acids when a new tRNA comes. So here was my tRNA that was on the last slide. A new tRNA arrives, and it brings with it an amino acid. Those amino acids that are next to each other form a bond. It's called a peptide bond. And the ribosome is then going to shift down the mRNA to the next codon. And when it does that, the tRNA that was previously there is going to leave. So you can see this first tRNA is leaving, and this ribosome's shifting to the right. It's going to keep working its way down the mRNA. So if you repeat those steps over and over and over, you're going to get a chain of amino acids. And that chain of amino acids is called a polypeptide. Eventually, the ribosome is going to reach a stop codon, a codon that tells the ribosome this is the end. You're not putting any more amino acids on the chain. It ends here. The chain ends. So when the ribosome reaches that stop codon, which in this case would be UAA, when it reaches that stop codon, it's done building this chain of amino acids. And that chain of amino acids, that's called a polypeptide. Okay, now, I showed you how a chain of amino acids gets built during translation. But you not only need to know the steps and how it's built, you also need to be able to identify which amino acids are going to be in the chain. And to do that, to determine which amino acids are in the chain, you have to use a codon chart. So a codon chart, there's a couple different versions, and I'm going to show you how to read both. In this first version, it's a table that looks like this. And essentially what you do is you just look up the codons that are on your mRNA. So these are the codons. You're going to look up these letters on the table and identify the amino acid that matches up. And so let me show you how to do that. Look at this first codon. Our first codon is AUG. So when I'm looking at this table, notice there were 3 letters in that codon. My first letter was A. So when you're reading this table, you're going to find the letter A on the left side. So there it is. Okay, my second letter was U. So you go up to the top and you find your second letter. So here is U. Well, oops. Okay, there's U. All right, then you look up your third letter over on the right side. So we're going to look for G. So my three letters were A, U, G. We found the A, we found the U. Now we're looking for the G. So here is my G. Now you'll notice there were more than one G on this right side. I chose this G because it's in line with the first letter. That's important. Whatever your third letter is, it needs to be in line with wherever your first letter was at. So AUG, where do all three of these letters connect on the table? They all connect right here at MET. So MET is shorthand for methionine, which is the amino acid. So what I'm going to write for the first amino acid is MET. So I looked up the first codon, and it coded for this amino acid, MET, which is short for methionine. So all these letters on the inner part of the table, they represent a shorthand for the name of an amino acid. So let's keep going. Let's look up the second codon for practice. Okay, so my second codon is GGC. So I would look on the left-hand side here. I see G. Up at the top for my second letter, G. On the right side for my third letter, C. And notice the C is in line with the first letter I picked. Where do all three connect? They all connect right here at gly. Gly is shorthand for glycine. So glycine was my second amino acid. So that's how you read this type of codon chart. But there's another one that you may find easier to read. It's a circle. So let me teach you how to read this one. In this alternative codon chart, you start in the center of the circle and you move to the outer edge. So the names of the amino acids are on the outer edge and the letters for your codons are on the inside. So you start at the center and you move out. So my third codon is AGU. So I'm going to start at the center, find the A, then the G, then the U. So you can actually draw a line to connect those if it helps. AGU leads to this amino acid. Ser is shorthand for serine. So I'm going to write SER for my third amino acid. All right, let's do another one to practice here. So my fourth codon is GUC. So I would start at the center, find the G, then the U, then the C. GUC, it leads to VAL, which stands for valine. So I'm going to write VAL right there. So these are the 4 amino acids that this mRNA codes for. Now, you can also look up this 5th codon. I'm going to go back to this chart for this one, UAA. So find the first letter on the left side of the table. There's the U. Second letter is A up at the top. Third letter, look up on the right, A. So where do all 3 of these connect? They all 3 connect at a stop. Now, stop is not the name of an amino acid. Stop means stop. You're no longer adding any more amino acids to this chain. So that's why I left this blank. I did not write the word stop here. I just stopped. I'm not adding any more amino acids to the chain. All right, so where exactly does this leave us? Well, by the end of translation, the cell has a polypeptide, and that polypeptide is a string of amino acids. So here are the string of amino acids that we built from the mRNA that we had on the last slide. We had MET, gly, ser, and val. And notice these are connected. Again, these bonds that hold the amino acids together, those are called peptide bonds. That's why we call this a polypeptide. It means there are many polypeptide bonds here. And so this chain of amino acids, that's what we have by the end of translation. Now, the polypeptide that is built is actually going to fold up into a specific shape. So you can see right here, we had a chain. It's going to fold up and take a specific shape. And once it has that specific shape, it can perform a specific function in the cell. And at that point, we have a protein. So, big picture, this one picture kind of sums it all up. Everything that happened inside the nucleus, that's going to be part of transcription. So that's where the mRNA got made. What we focused on in this video is once you have mRNA and it leaves the nucleus, what happens? So that's translation is going to happen outside the nucleus. It's going to happen in the ribosome. We're going to build a string of amino acids called a polypeptide based on the mRNA that we had. And once we have that string, it's going to fold up and take a specific shape, and it will be a protein. So that's how cells synthesize proteins.